Welcome to my short poster presentation about all the cool stuff in the latest stringApp version. I'm Nadezhda Doncheva from the University of Copenhagen. Why should you care about this tool? Nowadays, high-throughput technologies such as proteomics and transcriptomics produce long lists of genes or proteins that are associated with a certain phenotype of interest. This data usually is provided as a table like this one. In the first column we have some UniProt identifiers for the proteins, and in the last two columns we have log4 changes for two different treatments. A typical follow-up question is, what do these proteins have in common and what biology is behind the investigated phenotype? We can come closer to answering this question by using stringApp in Cytoscape to retrieve a network of functional associations between the proteins from the table, highlight the up and down regulated proteins from the experiment by using a red-blue gradient, as you can see in this network visualization, and we can also perform functional enrichment analysis in order to identify biological processes and pathways that are enriched in this specific list of proteins. On top of the already existing stringApp functionality, the latest version can perform cross-species queries. This means retrieving interactions for host pathogen or virus host pairs. Here you can see an example of a network that contains three different species, namely human proteins in teal color, proteins from the malaria parasite Plasmodium falciparum in red, and proteins from the mosquito that transmits malaria to humans in lime color. Furthermore, you can take any network loaded in Cytoscape that comes from a different database or from a protein interaction experiment and stringify this network in order to annotate the proteins in the network with information provided by stringApp. This information can then be explored in the new string results panel. Here you see how the panel looks for the nodes. You can, for example, filter nodes based on their subcellular compartment localization, or read the description of the protein and see the known protein structure. A similar panel is available for the edges, where the different string scores can be filtered, and also the network type can be changed to only show and retrieve physical interactions between the proteins and see if any of them are potentially participating in the same protein complex. Last but not least, one can also perform group-wise enrichment by looking on different clusters or other groups of the network and then exploring the results for those separately. I'll continue with an example of how stringify can be useful. Namely, you can start the analysis in Cytoscape with a list of interactions coming from an affinity purification mass spectrometry experiment like in this example. This is the supplementary table of the 2020 Nature paper where Gordon et al. Investigated potential physical interactions between SARS-CoV-2 proteins, which are the baits in this table, and human proteins, which are the preys. First, we imported the interactions from the table in Cytoscape. This is the resulting network in which each star formation represents one virus protein and the human proteins interacting with it. Next, we stringified the network using stringApp in order to retrieve different annotations such as the tissue expression, subcellular localization, and known protein structures of the human proteins. This also allowed us to add physical interactions from the string database in order to see if any of the human proteins that interact with different viral proteins actually participate in the same protein complex. We can see that there are quite some connections between the different groups, which means that this is actually the case. We also improved the visual style in order to better distinguish between the viral and human proteins. Here the viral proteins are these big red diamonds. Finally, we also performed functional enrichment analysis on all the human proteins by using genes mentioned in publications as gene sets. In this way, we identified a publication that studies the human proteins highlighted in yellow, and it's about how they are related to the nuclear pore complexes barrier, which is an important obstacle in how viruses enter the nucleus of host cells. If you want to try these new features and more, you can go to the Cytoscape App Store and add stringApp to your Cytoscape installation, or if you don't have Cytoscape, first install Cytoscape. If you have any questions or suggestions, you're welcome to come to my poster on Monday evening or contact me via email. Looking forward to seeing you all at ISMB soon.